In this session of virology, we're just going to cover an overview of human immunodeficiency virus, or HIV. A couple of important things that you can see in this image. It contains an envelope that surrounds its nucleic acids, and this envelope is important for gaining access into the cell, into a cell that's going to infect, such as a CD4 cell, such as a macrophage or a dendritic cell, for example. It contains RNA, so that's its nucleic acid. That's how it transmits its nucleic acid from one virus to the next, so to speak, is when it replicates. But it has important enzymes that allow it to convert that RNA into DNA, and that DNA can be incorporated into the host cell genome. So what we're going to do in this session is we're going to summarize how viruses are classified in a very simple way and explain the pathogenesis of HIV and briefly touch on the treatment using HAART. So how are viruses classified? Well, there are many different ways for classification, but in the end, the most popular one is based on one by David Baltimore. And this classification scheme is based on the relationship of the viral genome to its messenger RNA. Now, this is the scheme that you can see, and you can see there are 1, 2, 3, 4, 5, 6, 7 different classifications based on how the genome from the actual virus is turned into messenger RNA, because they all have to make messenger RNA because all viruses require the host ribosomes to make their DNA. So therefore, they have to make messenger RNA that can bind to ribosomes. So just as an example, you'll have some type 1 viruses that are double-stranded DNA. A type 2 is single-stranded. It then goes on to make double-stranded DNA and then directly makes messenger RNA. Just like in humans, we have double-stranded DNA, and we use an RNA polymerase to convert that into messenger RNA. So the viruses will use very similar enzymes. We have other viruses which are double-stranded RNA. We have some that are single-stranded RNA, and with single-stranded RNA, they can be positive sense or negative sense. Positive sense just means they're the same as messenger RNA, so they run in the same 5' to 3', and negative sense RNA just means they run the opposite way. And then finally, we've got another one here, which is your type 6. This is your positive sense single-stranded RNA, and this uses a DNA-RNA hybrid before making double-stranded DNA and then going on to make messenger RNA. And the reason I focus a little bit on this one, because this is what HIV does. And HIV has lots of its own enzymes that it expresses that allows it to do all this work. So, like I said, the basic classification is based on its DNA or RNA. That's the classification that's now used. Single-stranded RNA, double-stranded RNA, single-stranded DNA, double-stranded DNA, and positive sense and negative sense RNA. And so that's how it's actually classified. And so when we're thinking about HIV, there are two key ones, HIV-1 and HIV-2. They're part of the retroviridae group of viruses, so retroviruses. And as I mentioned, they are type 6. They're classified as type 6, and that's because they are positive sense single-stranded RNA that use an enzyme to convert that into double-stranded DNA eventually. That integrates into the host genome, and then that allows us to make messenger RNA and then proteins in assembly, for example. So that's a summary of how it can potentially go on to assemble and how it makes its messenger RNA. And I'm just going to mention 3 little key features because these are very important when we're thinking about treatment, but also just to show how clever this virus actually is. It has one enzyme called reverse transcriptase. Now, this allows it to convert its single-stranded RNA into single-stranded DNA. And so that makes it a very clever thing. Don't worry about tRNA, etc. So it's RNA to single-stranded DNA. And so now you've got an RNA single-stranded DNA kind of template, the duplex bound together. So they've got another enzyme, which is a ribonuclease, which degrades the RNA strand of that RNA-DNA hybrid. And it has a DNA, and then we have DNA polymerase, which allows it to copy that single-stranded DNA that's left to make double-stranded DNA. And it then has an integrase enzyme that allows it to integrate that double-stranded DNA into the host genome. Now, if you're thinking about HIV-1 and HIV-2, well, HIV-1 is worldwide, whereas HIV-2 is mainly restricted to West Africa. And it's important to note HIV-1 mutates very rapidly. So it takes only about a day and a half from when you get infected with HIV for it to infect the cells, make new viruses, and get released into the bloodstream. So you've got to act very, very quickly if you want to stop that replication process. But by the same token, it mutates a lot because some of these enzymes it uses don't have proofreading mechanisms. So they mutate a lot. So, like I said, we have that classification. We have HIV-1 and HIV-2, and then they're broken into different groups. And they're broken into the groups of Group M, which is major, Group N, which is new, for example, Cameroon, and O, which is an outlier, mainly in West Africa. And the M group is further divided into those subtypes. So, as you might expect, two viruses of different subtypes, so whether they're A, B, C, D, etc., they can infect the same cell type. And then what happens if you get infected with a type A and a type V, for example, you can get new progeny hybrid viruses with recombined genomes. And if these go on to infect more than one individual, they go on to form the CRFs, which are circulating recombinant forms. So recombinant versions of A, B, C, D, E, F, G, H, J, etc. Now, the three groups that we see there, M, N, and O, they may represent the three different introductions of SIV, simian immunodeficiency virus, into humans, which is where it's thought to have come from. Now, if we look at a little bit of history, AIDS was first described in 1981, so it's a reasonable time ago now. And opportunistic infections were first observed in young homosexual men, and they spotted things like Kaposi sarcoma, which was originally known as the gay cancer. Obviously, it's not, but that's a very rare skin cancer. They also seem to have pneumonia and obviously oral candida infections as well. So these initial observations suggested that there was some cell-mediated immune suppression, and that's why the syndrome was named gay-related immune deficiency initially. And this later became known as acquired immune deficiency syndrome, or AIDS. It was also noticed at the time that these individuals were more promiscuous, having unprotected sex in bathhouses, for example, and they were often drug users as well. And then people started to get these symptoms following blood transfusions, and that's when people started to think, maybe this isn't just a homosexual disease, and that's how it changed. So it's interesting to know a little bit about the history, about what people thought, and we'll touch on some of that history a little bit later as well. Now, there's also was AIDS denialism. So many people even now question the existence of HIV and even its links to AIDS. And for a long time, in particular in South Africa, where HIV and AIDS is most prominent in the world, this denial of the link between the two led to its ineffective response to the country's AIDS epidemic and over 330,000 early deaths. So how is HIV actually transmitted? Well, it's present in blood, semen, vaginal fluids, breast milk, in saliva and tears at much lower levels, and they don't tend to get passed that way. And transmission is obviously through anal and vaginal intercourse, sharing of needles, needle stick injuries, blood transfusions, not so much anymore, and congenital AIDS, so before or during birth or throughout breastfeeding. So even though it's present in saliva and tears at low levels, it's very, very rare to transmit HIV through kissing. Although that risk is increased slightly if they have sores, for example, in their mouth. Similarly, oral sex does pose a risk, as HIV-infected T cells from semen may enter abrasions or lesions in the mouth. So really, what's there isn't the issue. It's whether you have those open sores that is more of an issue. And for a long time, when it was first discovered, people were almost afraid some people were afraid to touch someone else for fear of getting HIV. Other people were scared of kissing people. So there was all these things that were going around quite a long time ago now, when there was a lot of ignorance and a lack of knowledge. HIV is also very fragile. Because it's got a lipid envelope, which is a plasma membrane, so it's made of lots of lipids, soap easily disrupts the virus because of that envelope. And HIV can't be transmitted by tears, sweat, or contact with mucus through coughing and sneezing, even though people did in the early stages think it could, hence some of the urban myths that were floating around for a long time. And mosquitoes also do not transmit HIV. So if we're looking at the prevalence, this image is a World Health Organization diagram showing how prevalent it is. Sorry, it's a little bit blurred, so it's hard to read. But you can see that in Africa is the highest prevalence, and then into the Americas it's more prevalent, and then into Europe. And then obviously you can see down in Australia, very few people are infected, so which is good news for people in Australia. Having said that, there are still about 27 to 30,000 people with HIV in Australia. So it's still relatively, a relatively high number of people. And when in that 2017 area, about 63% of HIV notifications were actually attributed to sexual contact between men. 25% were attributed to heterosexual sex, 5% to a combination of sexual contact between men and injecting drug use, 3% due to injecting drug use only, and then another 3% for a variety of other reasons. And it's interesting to note that HIV incidence actually is very low among female sex workers. So it's at around about 0.13%, so 0.13 people per 100 as of 2017. If we're thinking about the global numbers, since the beginning of the epidemic, over 70 million people have been infected with HIV, over 35 million of which have died. And globally, about 37 million people were living with HIV as of the end of 2017. Worldwide, between the ages of 15 and 49, about just under 1% of adults are living with it. Mind, the burden of that really is very, very varied, and it's mainly the African regions that remain mostly affected, with around about 4 to 5% living with HIV, and they account for about 2/3 of all people with HIV. So there are three main phases if we're looking at the phases of HIV infection. You can see there's the primary HIV-1 infection, followed by the chronic asymptomatic phase, and then if people aren't treated satisfactorily, it can move on to AIDS. So if you get this is your number of CD4 cells, so your CD4 count, because remember, HIV infects CD4 cells, which are your T helper cells. And so following an infection, so if you look at this primary infection, which is what you're seeing in this point, you can see in the earlier phases, you get the initial incubation period upon infection, and that's generally asymptomatic, and it usually lasts somewhere between 2 and 4 weeks. So you're not seeing any acute infection, symptoms at that point. You then hit the acute HIV infection stage. This lasts, as you can see, for an average of about 28 days at its peak, and you can get symptoms at that point, such as fever, swollen lymph nodes, pharyngitis, RAS, myalgia, malaise, and you can also get mouth and esophageal sores. You then move into that clinical latency phase, and this latency phase shows few symptoms, if any at all, and can last anywhere from 2 weeks to 20 years and beyond. Again, we're thinking about people who are untreated. However, there is still a massive turnover in your CD4 cells. They just get replaced at a similar rate. So you can see that whilst the CD4 count has dropped in those initial phases, during that latency phase, you can see they're still getting killed a lot, but they're being able to be replicated and get replaced at a similar point. But then when your HIV RNA copies, which is this thing in red, when your HIV starts to rise and your CD4 count starts to go significantly down, this is when the onset of symptoms of AIDS start to kick in, and you may get some of the symptoms of opportunistic infections. So what about the period? What about the primary HIV-1 infection period? So this is the period after infection, but before antibodies are detected. The virus replicates rapidly during this phase. The virus is very high numbers in the blood and your CNS and the lymphatics. It invades other tissues, but 50% of individuals don't have any symptoms. Those symptoms that people do have, like I've said, will be fever, sore throat, fatigue, rash, muscle pain, swelling of the lymph nodes, etc. And so this image is just on the bottom left, is just showing the HIV RNA load, so how much HIV RNA there is. And you can see that as time progresses, the HIV goes up. And you can see this increasing at this point. The symptoms will kick in at this point as well, and the antibody against HIV is kicking in afterwards. P24, that is just the capsid protein, so that's a protein that surrounds and protects the nucleic acid of the virus itself. So what's happening in that chronic asymptomatic phase? So there are no signs or symptoms of the disease, lasts in typical progressors, and we're going to talk about the different types of progressors right now. In the typical progressor, it lasts for about 10 years. So if we're just having a look at this diagram, the blue is viremia, so the amount of the virus in the blood. This is your CD4 count in green, so you can see the CD4 count drops initially, pulls back up, then you've got this 10-year period where it remains about the same before it slowly starts to go down. And you can see the immune response kicks up and then slowly deteriorates as your immune cells are infected and killed. So there are no signs or symptoms of the disease in this chronic asymptomatic phase of about 10 years in your typical progressors. Like I said, the virus continues to replicate, but the CD4 positive numbers are slowly dropping. And when these counts drop to less than 200 cells per microliter, that's not enough CD4 helper cells to help stimulate your immune system, AIDS and your opportunistic infections start to kick in. So we also have rapid progressors. So we have the typical progressors, we have the rapid progressors, and these hits off in about 10 to 20% of people. And they will generate or get AIDS within 2 to 3 years. So they're a lot more rapid. So again, if you have a look, you can see the viremia goes up very rapidly. You can see the immune response kicks in. But this time you can see the CD4 count drops much, much more rapidly. Therefore, you're seeing the immune response drop more rapidly. And therefore, you see the you see the viremia, the amount of virus, increasing much more rapidly. The reason for rapid progression isn't really known. Some people speculate that it may be due to some form of immunosuppression already present. Then we have our slow progressors. So this is, about another 5 to 15%, and they're free of AIDS for more than 15 years. And so again, if we're having a little look at this, you can see the viremia goes up, goes down, and goes down very low for an extended period of time. And whereas the CD4 count stays at a decent level for a decent amount of time, and therefore so does your immune response. But you can see over time, those slow progressors will kick off, and they will last a little bit longer. And then the most interesting group, I think, are the long-term non-progressors. And so 1% of individuals show no signs of the disease. So they have a normal CD4 count, T cell count, they have low virus levels, and they haven't taken any therapy. And this is thought to be due to some genetics. So they have what's called a chemokine co-receptor mutation. So this is just a mutation in the receptor for the HIV virus itself. And it's thought that in the mutations in this receptor, about 10% of Europeans are heterozygous. So they're classified as the slow progressors because they have got they're heterozygous as opposed to homozygous for the co-receptor mutation. Whereas 1% of Caucasians are homozygous, so they are the long-term non-progressors. The allele is absent in Asians, Africans, Indian, and Middle Eastern populations. Hence, they're not the slow progressors or the long-term non-progressors. And this mutation is thought to have arisen in around about the 14th century as resistance to smallpox outbreaks. So it seemed to help in resistance to smallpox. And smallpox epidemics were ongoing at that point, therefore allowing for natural selection. And this was part of that natural selection. The chemokine co-receptor mutations were allowing resistance to smallpox, but as it happens, they're also allowing resistance to HIV infection as well. So we've got these slow and non-progressors. So in other words, we've got a bit of HIV resistance. So like I said, these are mutations in the chemokine co-receptor. So this is our CD4 cell with a CD4 receptor, and then we've got a co-receptor, which is this chemokine co-receptor. So it's called CCR5, just so that you know what it is. And so what happens is HIV locks onto both CD4, which everyone's sitting with, and CCR5, and then it can get in and infect the cell. But of course, if you've got a CCR5 mutation such that HIV can no longer lock onto it, Your HIV floats around, it can bind to the CD4, but it can't bind to the CCR5 protein, and therefore it can't get into the cells. So individuals can resist infection, so they have HIV resistance. So what are some of the clinical symptoms of AIDS? So when you eventually go on to get AIDS, presuming you're not getting treatment, for example, without antiretroviral treatment, individuals tend to die within 2 to 3 years. And the death is generally down to opportunistic infections because of their decrease in CD4 T helper cell count. And some of those infections that people get will be cytomegalovirus infections leading to retinitis and pneumonitis. Variola zoster virus leading to shingles, herpes simplex virus 1 and 2 leading to cold sores and genital warts, and genital sores rather. Human papillomavirus leading to warts and cervical and anal cancers, and your hepatitis B and C viruses leading to hepatitis, obviously. You've got your bacterial infections such as tuberculosis, salmonella, pneumonia, fungal infections like thrush, and your parasitic toxoplasmosis and retinitis. So there are lots of potential microorganisms that can infect you and lead to opportunistic infections, in other words, leading to death. Another one, which is present in about 20% of AIDS patients, is your Kaposi sarcoma. And so this is this rare skin cancer that you can see in that image there. And AIDS encephalopathy is seen in about 2/3 of cases where they get impaired short-term memory and concentration, behavior changes such as depression, irritability, personality changes, and motor issues such as clumsiness, tremors, and leg weakness. So how do they test for HIV? Well, as a general rule, we'll measure your CD4 count and your viral RNA load. So if you get below 200 cells per microliter, that, as I've mentioned before, that basically defines clinical AIDS. So that's the point when you can no longer mount a significant enough immune response. And if you've got over 100,000 viral RNA copies per mil, that's also a very powerful predictor of progression towards AIDS. And so treatment is through HAART, so highly active antiretroviral therapy. And it's very, very powerful. It decreases the patient's total burden of HIV, helps to maintain the function of the immune system, and helps to prevent opportunistic infections that often lead to death. And in fact, treatment has been so successful that in many parts of the world, HIV has become a chronic condition in which progression to AIDS is increasingly rare. So, if we're looking at some of the treatments that are used in HAART, and at least 3 of the following are used during treatment, they may be non-nucleoside or nucleoside viral reverse transcriptase inhibitors. Now, if you remember, the reverse transcriptase inhibitors prevent the RNA virus being converted, or the RNA genome of the virus being converted into DNA. You've got protease inhibitors that are in there, and they inhibit some of its large proteins, they're called polyproteins, but some of its large proteins from being enzymatically cleaved into mature structural proteins of the virus. You've got some fusion inhibitors. So fusion inhibitors, obviously, they inhibit fusion of the HIV envelope, its membrane, with the cell membrane of the host cell, of the CD4 cell, for example. So they're blocking entry of HIV into cells. A GP41, that's just the fusion protein that mediates fusion. You've got integrase inhibitors. They inhibit the integration of the viral DNA into the host genome. And you've got the CCR5 blocking inhibitors, so the chemokine co-receptor blockers, again preventing docking onto the cells themselves. And of course, you would also have antivirals to prevent opportunistic infections as necessary. So, little question on HIV for you. So, which of the following are true regarding HIV? So, press pause, have a read through, and see what you know. Okay. Which of the following are true regarding HIV? HIV-2, but not HIV-1, can be transmitted via mosquitoes. That's incorrect. They cannot be transmitted via mosquitoes. Number 2, the initial period following incubation shows acute symptoms in approximately 50% of individuals. That's incorrect. As a general rule, there aren't any symptoms for the first 2 to 4 weeks, and following that, acute symptoms occur in about 50% of individuals. Number 3, inflammation due to HIV infection causes death in those who are untreated. That's incorrect. Death is generally down to the opportunistic infections that infect someone with AIDS. Number 4, there is significant CD4 cell death during the latent period. That is correct. There is significant cell death, significant CD4 positive turnover. However, cell division very closely matches cell death. Number 5, individuals who are slow progressors are homozygous for a mutation in the CCR5 co-receptor. That is also incorrect. The slow progressors are heterozygous for this mutation. The non-progressors are homozygous.